Hello, my name is Adam Packer, and I'm here to tell you about in vivo optogenetics. So first of all, what do we need in vivo optogenetics for? Why has it been developed and what is it used for? Well, our goal in using this technology is to manipulate neural circuits at a level at which they are constructed and organized. And what I mean by neural circuits is just groups of neurons that are acting together to perform computation. Now, we have a problem if we want to do this, if we want to manipulate neural circuits, and it is basically due to the fact that brains are big and densely packed. I don't need a scale bar for this image because it's a human brain. You all know the approximate size. It obviously has a lot of very complex gross anatomical features, which you can see even better if you take a slice of it and look at it end on. You can see all the sort of complex anatomy. And if you zoom in even on just the outer sheet, the neocortex of the brain, you can see this layered structure with different neurons of different types, different sizes, peppered throughout just this small chunk of the brain. And if you zoom in even further, if you use an electron microscope to go down to this sort of micron or nanoscale, you can see the individual processes of neurons are all overlapping in this space. So you've got processes of lots of different neurons embedded in even one small micron, 1/1000th of a millimeter of tissue might have bits of many different neurons in it. So we have a spatial problem. We have a difficulty of focusing our manipulation onto specific neurons in order to test how they might give rise to a particular function, how they act together as groups to perform a computation. And this is further confounded by the fact that there's dynamics, there's time, there's activity unfolding over time in brains. And so we can think of circuits as encoding and computing and decoding information over time. And just to give you an example of just how rich and complex these dynamics can be, I'm taking a famous example from the songbird literature. This is from Hanloser et al., Nature 2002. And what I've shown here is the song of a bird called the motif in this particular case. And what you're seeing is the frequency of sound over time. And these songbird songs are rich and complex, of different syllables like words that the bird puts together to make its song. And you can see they have complex dynamics, different frequencies over time. You've all heard bird songs, so I imagine you can hopefully picture what kinds of dynamics you might be seeing here on the screen. So if you record from different parts of the bird brain, you can find neurons that are active at different parts in the song. So for example, this neuron 2, which is shown here, has action potentials at this when the bird sings this part of the song in time. And in other parts of the brain, other types of neurons, there's different neurons that have different activity profiles. So each one of these ticks, again, is an action potential, spike of electrical activity generated in these neurons. These neurons, these interneurons in this particular case, it's a little more difficult to understand how their activity relates to the bird's song. But if you look at lots and lots of neurons in a particular part of the bird brain called the higher vocal center, or HVC, you can see that there's different neurons that are clocking out different parts of the song. So if you zoom in, for example, on this neuron, you can see on repeated trials of the song, it has very, very precise millisecond scale precision in terms of when it fires action potentials on each trial. So you can imagine if we want to manipulate these neurons, we're going to need a tool that works at this spatial temporal scale. We need to get ultimately down to individual neurons. We also need to have millisecond precision control in order to change the activity of these neurons and see what happens to the song and test our hypotheses about how these neurons give rise to the song. Here's another example using a different technology to expose the rich and varied dynamics of neocortical activity. This is from Chris Harvey, Nature 2012. And in this experiment, now in a mouse, they had an animal that was running on a virtual maze. They could present different cues to the animal on different mazes that they had made in a sort of virtual environment. And depending on the cues the animal received, it either need to go left or go right at the end of the maze. And it received a reward for getting the correct choice. The researchers then recorded neural activity from a particular part of the mouse brain and found that there were neurons that were left preferring neurons. So they had neural activity, that's what's shown here. They had neural activity that was again active at different parts of the trial. The x-axis here is time. And so the more red the raster plots here, the more active those neurons are. So you've got, and each row here is a different neuron. So you've got many neurons that are active at particular points on the trials where the animal will go left. These are the left preferring neurons. They're not active when the animal ends up making a rightward choice. And you've got neurons that are right preferring. They're active on right trials, but not left trials. And you can imagine doing more complex analyses on these, looking at the population level activity of these neurons. I'm not going to go into great detail here, but on different trials, you get different population level activity. So you can take this many dimensional activity of many, many neurons, and reduce it down to a picture that you can see in 3 dimensions of how the neural trajectory of activity unfolds for one trial versus the other. And you can see these different colors are for the different trial types, right and left. So again, we've got this very complex activity unfolding over lots of neurons with peppered throughout the cortical territory in this particular case with very precise timing. So we need a technique that will allow us to manipulate the activity of these neurons and test how they're encoding or computing the choices that the animal should make and ultimately the behavior and action that it takes. So how are we going to manipulate the activity of many neurons at once? Well, one thing we can do, and what has been very fruitful for researchers to do over a long time, is to inject electrical current via an electrode. And it's literally, as this picture shows, effectively stick a wire in the brain and apply an electrical current through it to bias the electrical activity of neurons near the electrode. And so this has been a very fruitful avenue of research. There's been very many landmark studies in this tradition, but it relies on the anatomical specificity. It essentially relies on putting your electrode in the right place, which you have to know beforehand what that is. So again, this has been incredibly useful. For example, here I'm showing a classic result from Dan Saltzman when he was in Bill Newsom's lab, showing, in this case, they had a monkey that was performing a behavioral task. And what it needed to do was make a behavioral choice that indicated the direction of motion of a bunch of dots that were moving throughout the visual field. So if all the dots were moving in the same direction, it was very easy for the animal to report that, and it got it right most of the time. But if the dots were all moving in a random direction, it had no way of knowing if there's any coherence to that motion, and it couldn't report that well. And without the stimulation, this open circles is the psychometric curve of the animal. And you can see that when they then applied stimulation using an electrode to the part of the monkey brain that processes motion, they were able to bias the performance of the animal, further proving that that particular part of the brain had an important role in helping the monkey determine the direction of motion of things in the visual field. So that's ultimately what we'd like to do with a sort of more precision, whether it's at the genetic level of the types of neurons, or maybe the spatial level of exactly where those neurons are, and possibly eventually even unpicking one neuron, but not a nearby neuron, based on what those neurons are actually doing, which is what I'll come to at the end of the lecture. So we need a method to manipulate circuits beyond what the electrode can provide, as useful as it has been. Now, Francis Crick formulated this most eloquently and concisely in a Scientific American article, now many years old, when he called for a method by which all neurons of just one type could be inactivated, leaving the others more or less unaltered. So he's focusing on inactivation, which is an important side of optogenetics, but you can also imagine the same for activation. And that is fundamentally what optogenetics provides. So what ultimately we'd like to have also is not just have this approach at the single cell level or maybe a circuit level, but all of the scales at which brains work. So from the synaptic level through the single cell and local circuit level to maybe even sort of interlayer organization, so activate one layer and look at the output in another layer of the brain, possibly even to entire brain areas. Now, I'll try to cover examples from most of these levels throughout the talk today. So over the years, there's been very many methods that have attempted to achieve this goal of changing activity now in genetically identified neurons. And that's important because there's different subtypes of neurons, excitatory neurons, interneurons, different subtypes of those. And in different brain areas, neurons' function can be ascribed based on their genetic identity alone. So there's been a large push to apply these tools in a genetic specific manner where you can, again, as Francis Crick said, activate or inactivate all the neurons of one type as at least identified by their genetic identity. So I list here many of the methods that have been proposed and used over the years to varying degrees of success. Many of them are still in use, but the one I'm going to talk about today, I'm going to focus on optogenetics with respect specifically to opsins. Okay, so opsins are light responsive proteins, have 7 transmembrane domains. There are 2 types. There's the microbial opsins and the animal opsins, or G protein coupled receptors. These are the ones that are in your eye, in the retina. And they all, both types, require retinol, which is this organic cofactor. This is the reason your parents always told you to eat your carrots when you were little, so that your eyes would work properly, so you have good eyesight. And the reason why is because the way these proteins actually work is that when the retinol that's bound inside them, and I'll show a diagram of this in a moment, when that retinol absorbs a photon, it isomerizes and triggers conformational changes throughout the protein. So the protein literally changes shape subtly to enable ions to flow across a membrane. Okay, so there's a lot. This is a very dense slide. Let me just give you the take-home points. These are light responsive proteins. There's 2 types of them, and they undergo conformational changes to allow ions to flow. So what do I mean by that? Well, first, let's talk about the microbial opsins, because these are the ones of the 2 types that are most widely used for in vivo optogenetics today. These opsins combine this light sensation, this conformational change after absorption of light, and ion flux, ions flowing across through the channel in these cases into a neuron that has this protein embedded in its membrane. So fortunately, even though these microbial opsins all require this bound retinol, there's a lot of retinol present throughout nearly all vertebrate tissues. So this strategy works particularly well because it can be used as a single component system to render cells light sensitive. All you need to do is express this one protein and you get the light sensation and the ion flux at once. You don't need to present any other cofactors or make the animal eat some sort of funny vitamin to make it work. It just works naturally with this one protein inserted into nearly any membrane. Okay, so I keep mentioning these proteins are inserted into membranes and they allow ions to flow. So let's get a picture of what actually happens here. I'm showing the sort of classic workhorse of optogenetics that started the optogenetic revolution in 2005 when it was first shown to work in mammalian tissues. And this is channelrhodopsin 2. And it is 7 transmembrane protein, like all opsins. It's a microbial opsin. It's actually a light-gated cation channel. And it being a channel is important in the sense that when it absorbs light, like any ion channel, it allows ions to flow into the cell. So if you embed this protein, shown in blue, into a membrane, a phospholipid bilayer, like on the the membrane of any cell, when you shine light on that cell, the ion channel will open. This retinol molecule inside the protein will change conformation slightly, allowing this pore to let ions into the cell. So if this is the outside of the cell, and this is the inside of the cell, you can have, in this case, cations rushing into the cell. So you've got positively charged ions rushing into the cell, and that causes a depolarization of the cell, which can ultimately lead to action potential generation in neurons. So to show what that looks like, here I've shown again this classic example from Ed Boyden when he was in Carl Deisseroth's labs in Nature Neuroscience paper 2005. And in this paper, they expressed channelrhodopsin in this case, as shown in this picture, cultured hippocampal neurons. And when you have enough of this protein expressed in this cell, and then you shine light on it, as shown by these bars here, you can generate enough current in those cells. You have enough cations rushing into the cells that you can get action potential generation in that neuron. And that's what's shown in the black trace here, is the electrophysiological record, voltage versus time, showing the spikes generated by the light. And now you may be thinking, okay, well, have we achieved the goal of sort of working at the spatial temporal scale that we think neural circuits work? Well, one thing we've achieved is not precisely shown here, but this has been quantified carefully in these papers and subsequent papers. We've got this millisecond precision. We can control exactly when the spikes happen. And we can also control in which neurons it happens by expressing the protein only in the neurons that we would like to activate. So to show an example of that, I'll show you one of the very first videos that really generated a lot of excitement in neuroscience in general, showing fiber optic optogenetic control of locomotion in a mouse expressing channelrhodopsin. So what you're going to see in this video is channelrhodopsin, which has been expressed in motor cortex of an animal. I believe it's on the right side, although we'll see when the video comes up. And then they activated with light using a fiber optic tether. They deliver the light to the animal remotely, essentially, just via a very small wire, basically, fiber optic. They deliver the light to the animal in vivo while it's in its cage, and then you can see the animal running in a circle because of the activation of motor cortex. And so what this showed is basically remote control of the correct neurons in the brain by activating. You can see the animal running around. You can see this hint of a wire here. And then they turn the blue light on, and the animal starts running to the left, I think, because they're activating right motor cortex. And so they're maybe driving more activity on the right side. So this is showing this sort of precise control. And another aspect of it as well, which is that it can be remote effectively, that with just a small tether to the animal, you can change the behavior of the animal in an ethological context, or at least nearly ethological context, in that it's mostly freely moving. Okay, now you can imagine that this is very exciting. You can turn neurons on. We have some control over space and time, and maybe which neurons we activate. Another thing we might want to do is turn neurons off. It's one thing to turn them on, but it's another thing maybe to show that they're sufficient for a given behavior. But we may also want to turn them off to show that they're necessary for a given behavior. So what I've shown here is halorhodopsin, which is slightly different to channelrhodopsin. It's light-gated still, of course, but in this time it's a chloride pump. So it's not a channel, it's a pump, which means it's a little less efficient, because a channel will allow ions to flow across the membrane according to their gradient, according to how many ions are on, what's the sort of pressure for the ions to flow across the membrane. A pump is a little bit different in the sense that it allows one cation or anion in this case to flow across the membrane per sort of photon absorbed. It's literally pumping it across one at a time. But in this case, it being chloride, a negatively charged ion, it causes hyperpolarization. It causes the neuron's voltage to go down, which leads to a suppressive effect in general on spiking, for example. So you can effectively turn neurons off or inactivate them. And that was shown in these papers, showing that if you are doing an electrophysiological recording from a neuron that's expressing halorhodopsin, and you're injecting spikes into it every so often just to monitor the electrical activity and show that it's spiking. Then when you turn the light on, so you flood the cell with light, activating the halorhodopsin, pumping chloride into the cell hyperpolarizes and no longer is able to reach action potential threshold, so you don't get those electrical events in the recording, proving that it works. It's also been used, I think, in a very exciting way to express this in human photoreceptors, which of course naturally hyperpolarize in response to light. And in this case, they've done it in a retina where the photoreceptor response was not intact, and they recovered it by putting halorhodopsin in the correct neurons and then applying light of the correct wavelength. I think this is really exciting and shows essentially the promise of optogenetic-based strategies for clinical applications. Obviously, in the first instance, most likely to happen in the context of blindness, but of course for very many other possible conditions as well, where one needs to manipulate the activity of a particular set of neurons in a particular way at a particular time. Okay, here's another interesting study enabled by optogenetics, now showing off sort of the spatial aspect of it, how it's useful to direct activation to different parts of an animal brain. And this is from an excellent study from Karel Svoboda's lab in Neuron 2014, Guo et al. And here, what the researchers first did is they had the animal trained on a task where it had to discriminate the location of an object in its whiskers. So rodents have a sort of tactile sense of the environment around them using their whiskers, much like cats. And in this case, they trained the animal to lick at a water spout on the right side if it felt a pole in its whisker field at this location, and lick left if it felt a pole in a different, more anterior location in its whisker field. What they then did was expressed channelrhodopsin in interneurons that tend to inactivate nearby neurons, and directed light, each one of these crosses shown here, across the mouse's neocortex. So this is the dorsal surface of the neocortex of a mouse. And they directed light to all of these different locations at different times when the animal was performing this behavioral task. So in this, for example, in this task, the animal was able to sample the location of the object in its whisker field for 1.3 seconds. And then it had a delay period of 1.3 seconds before it had to report its response by licking at a spout. And they inactivated all of these locations throughout the mouse brain during this sample period, and found that inhibition via optogenetic activation of interneurons of this part of the brain resulted in the biggest decrease in the behavioral performance of the animal. So they turn off this part of the brain, and the animal performs more poorly at reporting these locations. And this is particularly true when they do it during the period at which it's trying to sample that. Turns out this part of the brain is the somatosensory cortex, the chunk of brain in particular that's responsible for processing input from the whiskers. So this makes sense. They then also inhibited all of the same locations throughout the brain while the animal was waiting to report its response. So it's palpated the environment with its whiskers, it sort of whisked the surrounding area, figured out where they are, and then it has to wait before it can report its response. And during the waiting period, there's a different part of the brain that caused changes in performance most effectively. So this is the anterior lateral motor cortex, is the frontal cortex region, effectively, where the animal is sort of holding its belief, so to speak, about the answer of which way to lick. And so this is showing really the flow of information throughout the cortex at these different points in time throughout the task. And so it shows both the spatial power of optogenetics, that you can inhibit different parts of the brain, and it also shows the temporal power of it, that you can turn off different parts of the brain at different times with this, at least this level of the precision. And as I've mentioned earlier, even much greater than this. Okay, so I haven't really mentioned yet the key key part of optogenetics, of course, is the genetics part, that we can target neurons for activation or inactivation using their genetic identity. So we can hit a particular part of the brain at a particular time, but also only influence certain neurons and not other neurons nearby. Again, getting at Francis Crick's so eloquently and concisely described dream of turning one set of neurons on or off while leaving the others nearby unaltered. So this is shown by Aponte et al., Nature Neuroscience 2011, where they activated a particular subtype of neurons, the AGRP neurons in the hypothalamus, and showed that when they activate a large number of neurons, during that stimulation, they can cause the animal to eat more food. They found the neurons that appear to be sufficient to orchestrate feeding. And it's not just that there's a particular set of neurons, it's not just that it's this part of the brain. They had to hit particularly the AGRP neurons in that part of the brain, which they had genetic access to, meaning they could express opsin in only those particular neurons and not the other neurons also in the hypothalamus. And so it's that level of precision that really, I think, showcases, again, the utility of in vivo optogenetics and how it's helping us understand circuits. Another study that came out that year from David Anderson's lab, Lin et al., found an aggression locus also in the hypothalamus. So now hitting a different set of neurons. And I think this is just a really beautiful video where you've got the optogenetic tether activating a certain set of neurons in the hypothalamus. When they turn the light on, they can get the animal to attack even inanimate objects like this glove that they've inserted into the cage. And then they turn the light off and the animal goes back to normal, really showing that you can get quite complex and specific behaviors in a repeatable and remotely controllable fashion by having this technology that allows you to target activation to a particular set of neurons in a particular location at a particular time. Okay, there's another study that I'll highlight here from Susumu Tanagawa's lab. This is optogenetic stimulation of a hippocampal engram activates fear memory recall. This is basically saying they were able to reactivate a memory via optogenetic activation. Now that's a bold interpretation. What they actually did was they had the animal doing a freezing behavior. So they had previously conditioned the animal to freeze due to a fear of an upcoming electrical shock. And they were able to target their opsin to a particular set of hippocampal neurons that were activated during that fear conditioning, okay, so that they could later, when they weren't going to do, when the animals should not have been freezing, optogenetically reactivated the neurons that had been active during the freezing and it was sufficient to induce freezing behavior. Now, whether or not that is exactly and precisely a memory is, of course, up for debate and interpretation. But it's very interesting the way they did this. They tagged these active neurons via yet another clever way of labeling neurons using CFOS, which is an indirect marker of neural activity. So they could go back and say which neurons were active during a certain time period. So these studies have really highlighted, I think, the optogenetic revolution, a completely new way we're all doing neuroscience experiments because of in vivo optogenetics. So it was the method of the year in 2010. It gave rise to a breakthrough prize, which was worth $3 million in 2016. People say it's likely to give rise to a Nobel Prize in coming years. If you start searching for it on Google, it's one of the first things that comes up with the letter N. It's even got its own meme because, of course, everyone in lab meetings throughout neuroscience labs, they say, hey, have you tried optogenetics? And enough already. We've done it so much. But it just, I think, highlights how useful it is. As useful as it is, it still has limitations. It's not a perfect technique. So we know, for example, that the pumps that cause a lot of chloride to rush into the cell can cause problems. They change the GABA reversal potential for the GABA receptor, right? So this is what normally gives rise to inhibition because of the chloride gradient. But if you pump cells with a lot of chloride, well, strange things happen. Maybe that GABA receptor will now actually, when activated, cause chloride to rush out of the cell. And that can affect spiking later on. So it would change the electrical activity in a way that was not desired or expected. There's other pumps, such as proton pumps, which can cause spontaneous release from synaptic release sites. So there's issues, there's confounds. These techniques are not perfect. We have to remember we're putting in a protein that is not normally there, and we're pumping ions into cells that don't normally belong there, precisely to change their activity. And we need to be careful about the controls we do to confirm we're changing what we want and not what we don't. We can also express too much of these opsin proteins, which again are not endogenous. They're not normally there in the tissues we put them in neurons in the brain, for example. And so if you overexpress, there's methods, for example, like in utero electroporation, which can express lots of proteins to the point of overexpression. And this can cause abnormal axonal morphology. And many of the ways we express opsins are using AAVs, adeno-associated viruses, slightly different from in utero electroporation, but with sufficient titer of those viruses, they can also lead to overexpression that will also cause problems. So these are things we need to watch out for when we're trying to get opsins into our favorite chunk of tissue. There's also differences in the action potentials that are generated optogenetically. So if you, for example, evoke action potentials at axonal terminals, at boutons, at release sites, those axons, those action potentials that have not flowed down the axon may have different kinetics, resulting in differences in neurotransmitter release that one might be trying to study by putting opsin in those synapses. There's also off-target effects, and this has actually become a very big issue and conversation regarding optogenetics experiments in the last 5 or 10 years. Obviously, if we optogenetically activate a large number of neurons in the brain, it's likely that those neurons will have indirect effects on downstream circuits. And this complicates interpretation. If I photoactivate this part of the brain, and in fact, it has a big effect on some downstream region, and then I observe a change in behavior, it's hard to say whether that was due to the region I activated or the region that was subsequently activated downstream. There's also light targeting problems. So there's an issue. So light targeting problems is basically saying that light scattering is a problem, right? When we put light into the brain, just like if you try to shine light through your fingertip, the light doesn't go through all the way. You see this sort of diffuse light coming out the other side. And this is because, opaque things like tissue, brain tissue, they scatter light. They cause the light to be deflected from its course. And so that means that this, the genetic and anatomical specificity are great, but if we really want to get down to sort of levels at which we think circuits might function, where individual cells beyond their genetic identity might be working together as groups to perform some sort of computation, like the examples I showed you at the beginning of the lecture, we need to get around this light scattering problem. We need to be able to direct the light more precisely to areas that we want than by just flooding it through an overall region. So I've worked on some of these methods in my own work, and we can overcome some of these drawbacks of optogenetics by using something called two-photon optogenetics. So I don't have time to go into how two-photon excitation works, but as the name implies, it's a technique that requires 2 photons to be absorbed simultaneously by a molecule in order for it to change energy state or change conformational state. And this technique has an advantage in that it has a native sort of optical sectioning. You can only get these 2 photons to absorb simultaneously where the photon flux is very high in the focus of the light. So you can have a neuron expressing opsin, and you can scan a laser beam capable of 2-photon excitation across the cell body and generate spikes in that neuron via this approach, which allows you to target that individual neuron because of the inherent resolution and sectioning capabilities of 2-photon excitation. The 2-photon is a little less prone to scattering. We can go a little deeper in tissue, and this inherent sectioning gives us the ability to target individual neurons even in awake behaving animals. So just to show what this kind of thing looks like, we've also shown that you can combine this with a technique to split your light into 2 separate laser beamlets using something called spatial light modulation, which does exactly as the name sounds. It modulates light in space. So we generated 2 beams of light over 2 separate neurons here. We've scanned our two-photon excitation capable laser over each one simultaneously. And you can see in the electrophysiological recordings from each one of these neurons obtained via these patch clamp pipettes shown here, that each one of them spiked when we turned the light on. So we were able to activate these neurons without activating other neurons nearby. So if we move these spots of light off these cells, for example, we see that they're not activated, showing the specificity of this sort of approach to overcome some of the drawbacks of optogenetics. Another thing we'd really like to do is, as I've been saying throughout this, what we want to know is how activity in neural circuits gives rise to perception and action. And so what we'd like to be able to do is have an animal performing a behavior, monitor the activity of identified neurons that we believe underlie that behavior, and then close the loop in that particular animal by manipulating the activity of those identified neurons to test our hypotheses about how they underlie that behavior. So we want to actually combine the reading and writing of neural activity in vivo. So obviously the optogenetics I've just described gives us the ability to manipulate activity with this sort of high spatial temporal precision. We also need to have a technology for reading neural activity in order to sort of combine these reading and writing channels in vivo. Now, in order to do that, we combine two-photon calcium imaging, which I haven't described today, but hopefully you'll be able to get that from another lecture. Effectively, what this enables us to do is monitor the neural activity of a population of neurons using a calcium indicator of neural activity. So shown here is GCAMP6S. This is a genetically encoded calcium indicator. It's expressed in all of these neurons in green. So each one of these circles is over a neuron expressing either both GCAMP for reading neural activity and C1V1 and another opsin for writing neural activity. And what we've shown in this proof of principle experiment in this paper published now a few years ago is that we can record the activity of neurons and also stimulate them. So for example, we can identify neurons in the mouse barrel cortex. This is again the part of the rodent, the mouse brain that's responsive to tactile stimulation of the whiskers. And we can find neurons shown by the squares that are active when we flick the whiskers in one direction. And we can find neurons shown by the triangles, which are active when we flick the whiskers in another direction. So this neural activity recording is in units of fluorescence versus time, but it's basically showing it's an indirect readout of the neural activity in these neurons in response to the sensory stimulation. There's also neurons out there that don't respond to either of these particular stimulations. Presumably they respond to some other sensory stimulation. And what we've shown here is that we can then, using two-photon optogenetics and our spatial light modulator, direct photostimulation to the neurons, all of the square neurons as a group. We can photostimulate those neurons, and so that's shown in the calcium imaging record here as this pink vertical bar. And what you see is over time, when we turn on the photostimulation, we get robust activity in those neurons that we targeted here. And then we can do the same for the other groups of neurons, the ones that respond to the other flick, and so on. And so, of course, that begs the big question. What did the animal feel when we photostimulated the groups of neurons that normally underlie the sensory response to this particular stimulation? Did the animal, for example, feel a sort of virtual or surrogate sensation as if its whiskers were being flicked? And the answer is, ultimately, we are able to bias the performance of animals if we activate enough neurons. So we've got a preprint on bioRxiv now that we hope is coming out soon. And hopefully we'll be able to link that to the lecture here, or you can find it online if you search for my name. But we've ultimately been able to bias the performance of animals, not only by using the power of in vivo optogenetics to target activation to the correct neurons, but also by doing it in the correct, when the animals are in the correct state and controlling for sort of the behavioral variables to make sure it's the neurons that we are targeting in particular that are having this effect. So with that, I'd like to close and thank you for your attention.